Hey guys, welcome back. I want to ask you to stop the video quickly and try and answer the question that we have on this slide. If you don't manage yet to answer this question, don't worry about it, and we're going to find out how to answer it together in this video. So today we're going to cover the first bit of the 15% of seeing and hearing. So specifically, we're gonna see how do we sense, perceive, and see the world. Few important elements and terms also that you guys have to remember for the exam are listed on the slide. Specifically, quite important are retina, optic nerve, fovea, and blind spot. So before diving into them, I think all of us sort of have an idea of what happens in our brain for us to have the perception of the visual world, right, that we perceive every day. How it works is basically that light falls through the pupil into the eye, and this light interacts with the retina, and the retina is a light sensitive surface. Also, this surface is filled with photoreceptors that are stimulated by the light, and this stimulation leads to the images that we perceive. Photoreceptors, together with retinal neurons, have different functions. They convert light into action potentials. They allows us to distinguish between the different colors because they allow us to distinguish the different wavelengths. And also, they enable us to distinguish the different light intensities. Also, fun fact, and a fact that is important to remember, is that the image that is projected onto the retina is projected upside down and backwards, and then our brain changes the images so that they are oriented normally again. Then we get to the fovea, and basically what happens is that our vision is sharper at the center of our visual field, because rather than in the periphery, because in the fovea, so the center of the retina, the photoreceptors are closer together. Then we have the blind spot, also called optic disc, and this element, this region, is called like this because there are no photoreceptors. And the way we compensate for this blind spot is that the visual field of the right eye and the visual field of the left eye overlap. And so we do not perceive the blindness of the spot. Then we get to the photoreceptors. The photoreceptors convert light into first chemical energy and then into neural activity, which leads basically to a change in the release of neurotransmitters. We have two types of photoreceptors, as you can see on the slide. We have rods and cones. Very important that you remember the differences between these two types. Rods are very sensitive to low levels of brightness, so they are like specialized in night vision. They cannot see color. They have all the same light absorbing pigment, and that allows us to also see the movements. Cones, on the other hand, are very sensitive to bright light and colors. They allows us to see fine details, and they have three pigments, which are blue, green, and red. Now, remember that we said that we have our sharpest vision in the fovea. Well, that is because we have way more cones in the fovea rather than outside. Now also, we are less sensitive to blue wavelength than to red or green, because the blue cones are less compared to the green and red ones. Then we have the retinal ganglion cells, which are retinal neurons. There are two types, two layers, to which photoreceptors are connected. The first layer consists of three types of cells: bipolar, horizontal, and amacrine. The horizontal cells connect the photoreceptors to the bipolar cells. The amacrine cells connect the bipolar cells to the second layer of the retinal neurons, which are the retinal ganglion cells that are subdivided into two types. The magnocellular cells, which are sensitive to light. They get more information through the rods, so this means that they are not sensitive to color and are found throughout the retina. And then we have the parvocellular cells, which are sensitive to color, which means they receive information from the cones, and they're mostly concentrated in the fovea. Okay. Then we get to briefly talk about the receptive fields. It's important for us to know that every sensory receptor has a receptive field, which is a part in our body sensitive to a sensory perception, right? What happens here is that we have a comparison of information between different receptors, and the overlap between the receptive field allows us to see events. Then we have the ganglion receptive fields, which is the region of the retina over which stimuli change the cell's firing. It is vital for you to remember that there are two types of cells involved. There are the on-center ganglion cells and off-center ganglion cells. The on-center cells, when light enters the central part of the ganglion cells, right, there is the excitation of the cells. Whereas when light falls onto the off-center receptive field of the ganglion cells, the cell is inhibited. When the light falls all around the receptive field of the cell, the firing increases weakly. Outside of the center of the cell works in exactly the opposite way. So in the off-center cells, when the light enters the center, then there will be inhibition, right, of the cell. Whereas when the light will hit the out part of the off-center ganglion cells, then there will be excitation. So now stop again the video, please, and try to answer the question that we saw at the beginning of the video. We now do have the instruments to answer it. And for any questions, remember that we have the WhatsApp group where I'm always available. And yeah, we cover a tiny part of the seeing and hearing topic of brain and cognition. And hopefully you enjoyed the video, and see you in the next one.